Today we're talking about generic medications. Generics are an additional brand of an existing medicine that are taken with the same active ingredient. You're only able to make generic medications once the patent of the original drug manufacturer has expired. Pharmacists legally are allowed to substitute the brand name for the generic brand without referring to a prescriber, unless the prescriber has indicated on the script that it's not permitted. But they do need the permission of the patient. This means that it is a pharmacist's job to advise the patient on the safety and suitability of the generic. Generics, unlike skincare and makeup dupes, are highly regulated. They cannot just be made with the same active ingredient, they have to be deemed acceptably comparable by a regulatory body. A drug that is deemed bioequivalent provides the same plasma concentration of the same active ingredient and has been assessed to have the same safety, effectiveness, physiochemical, biological, and immunological characteristics as the original name brand. The bioequivalence information of the brand name and generic is provided for the pharmacist by the PBS. We see here on the PBS that all of these drugs are bioequivalent, as indicated by them having the pink A next to them. Some brands are going to be flagged by the PBS as not having enough evidence to determine if they are equivalent. This doesn't necessarily mean that anything is wrong with taking these medications. Instead, it just means that more caution should be taken when substituting. This is likely going to involve assessing patient risk factors, the patient's financial ability to buy the brand name, the therapeutic index of the drug, and advising the patient about a lack of evidence. In the case that a prescriber has marked that their brand cannot be switched, but there's no name brand available in the pharmacy, the pharmacist can consult the prescriber, or they can simply get the patient to consult the prescriber. A patient must give informed consent, and it is the role of the pharmacist to provide information. A patient can then make a decision based on the cost, formulation, route of administration, certain allergies to excipients, other medications they're on, and their trust of the brand. Part of informed consent is also making the patient know that there is a generic option before the drug is dispensed. But for long-term therapies, we're going to be using the same brand the entire time. Some things to look out for as they might lead to problems when there's substitution is drugs that have a narrow therapeutic index, or epilepsy medications, as well as patients that are at risk of confusion, like patients who have cognitive or visual impairments, patients with complex medical plans, or patients that have English as a second language. We also want to look out for patients who have mental health conditions, where if the brand name is changed, it might have an effect on adherence. We also want to keep out for lactose and gluten as excipients, since they are allergens, and also gelatin as an excipient, since it is haram and can't be had by people who are Muslim. The TGA says that drugs are bioequivalent if the bioavailabilities are so similar that for the same dose, their efficacy and safety profile are the same. The TGA must be provided with data that demonstrates interchangeability between the brand name and the generic. This comes from pharmacokinetic studies which prove bioequivalence. In these studies, a group of volunteers are given the original medicine and then on a different day, a generic medicine. Blood samples are taken at set intervals after dosing and the rate and extent of absorption is assessed. We also look out for any adverse effects that are seen. Typically, these studies are single-dose studies which are not blinded, but we do randomize the order that the volunteers take their doses. We also don't use a placebo. Instead, the reference is the brand name medication. The volunteers for these studies need to be 20 healthy young males that receive a low oral dose. These volunteers must meet diet, exercise, alcohol, and medication requirements, and this is to lower the variability between samples. There is also a standardized BMI, and normally fasting is required before the trial. In the case that it is a medication that needs to be taken with food, a standardized meal is given which has a known nutrient profile. We take blood samples for 7 half-lives and take around 16 to 24 samples. Often we measure drug concentrations using high-performance liquid chromatography, which is highly specific and selective. We take 3 parameters from these studies, the Cmax, the Tmax, and the area under the curve to infinity. The area under the curve is the most important of these parameters since it demonstrates the extent of absorption and the systemic exposure to the drug. Sometimes a manufacturer wants to make a new generic that is at a higher dose than the name brand is. To get this approved, a bioequivalence study needs to be conducted, but we must also analyze this information to determine the relative availability of the generic compared to the market leader. To do this, we must normalize the area under the curves to make them relative to the dose and then compare them. And we do that with this equation. Here we take the area under the curves and we divide them by the doses to normalize them. Then we divide the generic by the reference. In this equation, we are assuming that the clearance isn't changing between the products and that the drug has linear pharmacokinetics. When we fill in this equation, we should get a percentage in the form of a decimal. So if the bioavailability is 1.17, it also is 117% similar. For a drug to be deemed bioequivalent, it must fall within 0.8 and 1.25. As you notice, this is a range that is skewed a little to the right. It isn't exactly 20% on either side. This is an asymmetric confidence interval, and this is because of a little quirk of pharmacogenetics. Genetic mutations in the genes that code for metabolic enzymes is not very uncommon. This means that when we're plotting the area under the curve for a large population, we're going to see that there is right skewing in the area under the curve values. This is because there are less metabolic enzymes in that population that has the genetic quirk, and therefore it's harder for them to clear the drug, leading to a larger area under the curve. It's also worth noting that for drugs with narrower therapeutic indexes, there tends to be a narrower acceptability criteria. Bioequivalence tests are great, but they're not without their own flaws. One of these flaws is that for some drugs, the area under the curve is not the most important parameter to be considered. So for example, when it comes to drugs that are administered in emergency situations, like in overdose, it's important that the Tmax is very, very small, since the drug needs to be effective fast, but this isn't considered in a bioequivalence study. These tests are also only conducted on healthy males, and so it doesn't provide any understanding on the effectiveness of the drug on variable physiology and the actual practical aspects of pharmacokinetics.